Good afternoon, ladies and gentlemen. Welcome to this session on extracellular vesicles and nanomedicine. We have, I think, a couple of very interesting lectures ahead of you. I don't want to spend too much time on the introduction because, as you know, it's 9 minutes at Klinam in Basel, so it's only eating away of my own presentation because I'm actually the first presenter. So I suggest that we start. My name is Raymond Schiffelers. I'm a professor of nanomedicine at the University Medical Center in Utrecht, and we investigate lipid nanoparticles, so synthetic particles, as well as extracellular vesicles for RNA delivery. The attractive part, of course, why we study extracellular vesicles is that they could be natural nano-sized drug delivery systems. They contain essentially all molecules that can be present inside and on cells, and they can transport them over a distance to acceptor cells. And as a result, they've kept found the imagination of many scientists that they could be carriers of nucleic acids, for example, between cells. The difficulty, of course, in studying what happens after a vesicle actually arrives in an acceptor cell is the fact that it contains thousands of bioactive compounds, including the RNA of interest. So it is very difficult to really understand how vesicles achieve their delivery function and how efficient they are actually. So we set out to really investigate that in more detail, and for that we developed this CRISPR-Cas-based reporter system for single-cell detection of EV-mediated functional transfer of RNAs. And these are the important people involved. Olivier de Jong developed the assays, Dan Murphy did a lot of the assays and comparisons with the lipid nanoparticles, and Peter Vader was responsible for the design of the experimental work. This is how the CRISPR-Cas reporter is built up. It contains a promoter and an M-cherry production, so a red fluorescent protein. And then we have a linker, and that is a target for Cas9 single guide RNA. And the moment that the Cas9 actually is activated and the cleavage is made, you can get 1 or 2 nucleotide frame shifts that result in the correction of an out-of-frame green fluorescent protein. So what happens if the single guide RNA activates Cas9, then you actually get a color change from red to green inside the cell. And you can see that illustrated below, the concept that we're investigating, panel D. So single guide RNAs are packaged in EVs and then would activate the stoplight expression, and then actually the cell would turn from red to green. And well, we can see that we can achieve that over here if we transfect the single guide RNA with a synthetic lipid nanoparticle. You see green cells appearing. And also if we co-culture these cells, we see green cells appearing. And what of course makes sense is if you have more donor cells, then more receptor cells will actually turn green. They also do that without cell-cell contact. So that could be then the extracellular vesicles that are mediating this. So here you see the untreated cells. The non-target single guide RNA doesn't activate the green fluorescent protein production, but the target single guide RNA does. So you see these green cells appearing, and they are sensitive to uptake inhibitors like GW4869. Also, we can isolate the extracellular vesicles first and add them to cells, and then still we see the green cells appearing. So first of all, we isolated them with size exclusion chromatography. They're about 100 nanometers in size and contain all the markers that you would expect for bona fide extracellular vesicles. This is how they look under the electron microscope. They contain the RNA of interest, and only after you disrupt vesicle integrity by adding a surfactant like SDS, then you see the RNA disappearing. The vesicles are taken up by cells, as you can see here with fluorescently labeled EVs. And finally, also green cells start to appear if you add them to cells in culture. They're dependent on specific uptake pathways. So if we knock out certain pathways in the producing cell or certain pathways in the receptor cell, then actually the number of green cells that appear are strongly reduced. So we can see which pathways are involved in the trafficking of extracellular vesicles. Same holds true if we add them. So we isolate the EVs first and then add them to cells in which certain pathways have been inhibited. then we can also see a very clear dependence on certain pathways, such as, for example, the covalent pathway. In this case, adding the covalent-1 siRNA strongly reduces the number of green cells. So now for the functional comparison, how can we compare such an EV to a synthetic lipid nanoparticle in which you have the single guide RNA loaded? First of all, we made a dose titration on when we saw the extracellular vesicle effect kick in. And that was, in this case, somewhere between the 10 and 40 microliters of EVs that we added in the MDA cells, and also in the A431 cells. We see that between 10 and 40 microliters, we start seeing more and more green cells. And we tried to estimate how much single guide RNA was in there. So we made a standard line, and then we arrived at somewhere around a 1 femtomolar amount of in 10 to the 5th extracellular vesicles. And then we compared that to the ability of synthetic lipid nanoparticles to actually achieve the same. So whereas the EV concentration range is over here, so at a very low concentration range, we already saw the effect kicking in. So that is between the 10 and 40 microliters of EVs that we add. We see that we are about 100 to 1,000-fold more efficient compared to the state-of-the-art synthetic lipid nanoparticles based on the MC3 lipid, so that is the Alnylam formulation. So what we've shown is highly efficient transfer of single guide RNAs by EVs, about 100 to 1,000-fold better than state-of-the-art LNPs. But of course, the loading is the challenge because we're working in the femtomolar range, whereas with synthetic LNPs we can easily work up to micromoles or millimoles range. So that is still one of the biggest challenges, I think, for the EV field regarding RNA delivery. And with that, I want to thank the funders, ERC for Peter, and in the past for myself, Horizon 2020 projects, and also the Netherlands Organization for Scientific Research. So with that, I want to stop sharing this presentation, and we're back, and I can also check right away whether there are any questions, and I have to go here. I see that there are no questions up to now. So that means that we are very well ahead of time, actually. Questions perhaps from one of the panel members? Questions online? No, then we can prepare for the next presentation. Ivan, working at Valdebron, responsible for the preclinical research and functional validation. One of the last places I visited before we went into lockdown. It was a very enjoyable experience. So take it away.
Okay, thank you very much, Rai, for the nice introduction. So I'm really happy to be here. I would like to thank the organizers to inviting me here and to enable this whole session about the extracellular vesicles. Sorry, this is working by itself. So I said, Raymond said, my name is Ivan Elasola. I'm coming from the Valdebron Hospital, which is a tertiary hospital in Barcelona, in Catalonia, and as that is receiving many patients with rare diseases, but it has a long history of working with rare diseases in research, and this is what I would like to introduce to you now. No? So as a starting point, I will give you just 2 slides on what are lysosomal stress disorders and what has been done so far using extracellular vesicles in the literature for treating these diseases. And then I will show you our results with dealing with Fabry disease, is one of these type of lysosomal stress disorders. We were working with extracellular vesicles containing this protein, the GLA, and we have in vitro and in vivo results that I would like to show to you. And then we also move into a different disease, which is the Sanfilippo A, dealing also with extracellular vesicles. So it seems that my PowerPoint presentation is moving by itself, so if there is a problem with that, just let me know. But coming to the definition of the lysosomal stress disorders, this is a group of diseases, actually. All of them are caused by the deficiency of the alteration of lysosomal enzymes. It could be also a deficiency or an alteration on a transporter. In any case, this is a group of diseases, many different diseases. Each of them individually is a rare disease, but if we take the whole group, as you can see here, the incidence raised up to 1 every 5 to 10,000 individuals. So it's pretty relevant. The most beautiful thing of the lysosomal storage disorders is that in many cases it's the pediatric population that is affected, and they usually die at very early ages. This is not the case of Fabry disease. It's one of the most prevalent disorders, lysosomal storage disorders, but in this case, patients arrive to adult stages. The disease in this case is caused by mutations in an enzyme that is named alpha-galactosidase A, GLA. Is coded in the X chromosome. It means that all males are affected, but female carriers also have symptoms of the disease, and they are also being treated. So it's important to know that. And what happens when we have a mutation in this enzyme? It happens that we get the accumulation of the substrate of the enzyme, mainly the GB3, this globotriacylceramide, because the enzyme is not able to hydrolyze this bound, and the whole substrate gets accumulated inside the lysosomes of the cells. This is a multisystemic disease because we have lysosomes everywhere, but the mainly affected organ is the kidney. Patients that do not go into treatment, they usually go into dialysis very early and kidney transplantation. Main organs, as I said, is the kidney, but heart, brain, gastrointestinal tract, and skin are also affected. So currently there are 3 treatments approved for the treatment of Fabry disease. 2 of them are based in the administration of the naked enzyme. The recombinant enzyme is what we know as enzyme replacement therapy. And this one in here, Replagal or algasidase alpha, is the one that we are using as internal control in our experiments. Different treatment is the use of chaperones, in this case migalastat, that is only available for those patients that had a residual activity of the enzyme, so for certain mutation, not available for all Fabry patients. And in clinical trials, we already have different types of gene therapy, ex vivo or in vivo, and also the substrate reduction therapy here. So the use of enzymes or the enzyme replacement therapy in these pathologies is based on a mechanism that is well known in natural cells. So we have the synthesis of lysosomal proteins in the endoplasmatic reticulum. They go into the Golgi. In the process, they get glycosylated, and then with the training reaction in the transfer from the ER to the Golgi, these mannose residues are exposed. They are they add the phosphorylation here, and they are able to bind the receptor in the Golgi and go to the lysosome. But up to 20% of the protein that is produced in this process is not reaching the lysosome, is being secreted to the extracellular media. And from there, the same cell can either take it back to the binding or through this membrane mannose receptor, or it can be available for a different cell. And this is actually the base of the enzyme replacement therapy. You put there a bunch of a huge quantity of the recombinant protein, and if it's properly glycosylated and if you have enough mannose receptors, you can get the protein in your cells. But as you can envision here, this treatment has problems in the sense that it's very poorly available to the cells, and the intravenous administration of the enzymes have to be repeated every 2 weeks. The half-life of the enzyme is really low, and they do not reach all the organs in the required quantities. So in this scenario, nanotechnology and nanomedicine was seen as a way to overcome these problems. Why? Because these vehicles are able to protect the enzymes from the degradation of the generation of the autoantibodies. We also avoid the internalization through the mannose phosphate receptor, and we can improve the biodistribution. And actually, if you are able also to put specific targeting moieties on the surface of these vehicles, you will be able to cross the blood-brain barrier. And indeed, this was the objective of one of the European projects where we participated, the Smart for Fabry. This was a project that I already presented in Clean years ago. But I would like to take the chance here to tell you that we are having really good results in terms of efficacy using experimental models in vivo. We have already almost concluded the preclinical regulatory testing, and the orphan drug designation for this liposomal GLA system is already being asked to the EMA. So, but today was not the day to talk about the liposomes. It was the day to talk about the extracellular vesicles, the natural counterparts of these liposomes, and the advantages of these systems are that they are biocompatible, biodegradable, they show low toxicity, low immunogenicity, they can be stable in different fluids, and they can be easily generated. And in this sense, we have different approaches. We can use bottom-up approaches where we engineer the cells to produce the extracellular vesicles with the cargo already inside them, or you can use top-down approaches by loading mechanically or chemically the cargo inside the extracellular vesicles. So first, in this case, you first purify the extracellular vesicles and then you load them. And regarding the use of these extracellular vesicles as vehicles for the enzyme replacement therapy in lysosomal steroid disorder, there are just two examples in the literature. One of them from the hands of the group of Bratakova and Japer Hill, where they used these top-down approaches, loading extracellular vesicles with the TPP1 protein for treating Batten disease. And the second example is the use of HEC-derived extracellular vesicles with the bottom-up approaches done by this group in here for Gaucher disease. In our case also, we choose to work with the bottom-up approach. So we were introducing the expression vectors coding for the protein inside the cells, either transiently or constitutively. In this case of the Fabry disease, we choose to add a tag moiety to the protein, so we were able not just to have the extracellular vesicles, but also purify the very same protein from the media, let's say the soluble counterpart of this, what's been inside these extracellular vesicles. And this gave us a very good comparator to be able to conduct all the in vitro and in vivo experiments. So before going ahead and showing you the results, I just like to remind you that when we are talking about extracellular vesicles, actually we are mixing different things, and we are seeing different things, not just in size, but also in the origin of the vesicles. We have the apoptotic bodies that are produced during this apoptotic process, and they're very big. So in the fractions that we are isolating, we don't have presence of this type of vesicles, but we do have a mixture of microvesicles and exosomes when we are isolating by the methods that we are using the extracellular vesicles, right? So the difference among them is not just the size, it's also the origin, how they are produced. And in the case of the exosome, which is the population that we have mostly represented in our products, they are originated from the fusion of the these NVVs with the plasma membrane. So in some extent they have a common origin with the lysosome, and maybe this is one of the reasons why we have this increase in the quantity of the lysosomal proteins and activity inside these extracellular vesicles. But let's get to that now, because what we did was
you need to round up.
Sorry?
You need to round up, I'm afraid.
Okay. So I need to finish?
Yes, please.
I thought I had 15 minutes, sorry. So what we did was to isolate these extracellular vesicles. We saw that they were containing mainly exosomes, very high quantities of the GLA protein and other lysosomal proteins. In vitro, we saw that they were colocalizing with the lysosomes, and they were reaching high enzymatic activity in the samples that we are isolating, and in vitro, much more activity than the free enzyme. So then we labeled the extracellular vesicles with a fluorochrome. We saw that they were reaching different organs, more or less with expected biodistribution of a nanoparticle. We could see that it was not just the fluorescence, we could also see the protein there, so everything was traveling together. Reduction on the GB3 levels, which is the substrate of the enzyme, and we could see really reduction also by measuring the GB3 levels by mass spectroscopy. And this was done in different tissues. This is in the case of the kidneys, but we did also the experiments with the repeated dose administration. We saw the same results in the kidneys, high activity for the extracellular vesicles, very much higher than compared to the free enzyme. But the interesting thing also in here, and let me go very quickly on this, is that when we were administering intravenously the extracellular vesicles labeled with the DAR fluorochrome, we were able to see that also in the brain parenchyma. Not just that, we also saw a reduction in the GB3 levels, we were not reaching the wild type situation, but we were much more closer than with the free enzyme, which is not able to cross the blood-brain barrier. So we were able to cross the blood-brain barrier and have efficacy also in the brain. And moved by these good results, we saw that the same thing could be applied for different lysosomal storage disorders, such as the Sanfilippo. So just to finish, because I think that I'm out of time already. This was just to show you that extracellular vesicles can be a good vehicle for lysosomal proteins, specifically enzymes, because we can increase the activity. I haven't shown that, but we were able to lyophilize these extracellular vesicles and see that they could maintain the activity. And in Fabry disease, we are having very good results that we are extending now to other pathologies. I'm really sorry that I thought that I had 15 minutes and it was not the case. So thank you very much for your time. And I will be glad to take questions at the end of the session.
Perfect. Thanks a lot. Indeed,
thanks for letting me know that I was late.
Yeah, so. No, it's fine. Perfect. Thanks a lot. We'll indeed go to the discussion straight at the end. And now it's time for Mike. He has been able to solve the technical issues. So Mike Keller from Roche in Basel will tell us about the milk exosomes that they've been working on.
Okay, excellent. I think the technology works now. I hope you can hear me all right. Good afternoon. Sorry for these technical problems that you had. My name is Michael Keller, and over the next couple of minutes, I would like to present you some work that we did in the frame of a postdoc project, which dealt with the investigation of extracellular vesicles as carriers of locked nucleic acid antisense oligonucleotides, which we call LNAs. The reason that we got into that field was some scientific literature that reported that extracellular vesicles are orally bioavailable and reportedly even had the capacity to transport exogenous oligonucleotides to the brain after oral or IV administration. Now it is established in the literature that EVs are naturally occurring transport systems for cargoes over short distances. Now whether they also can travel over longer transport distances, including to the periphery, after oral administration is much less certain. Now, in our hands, we focused on EVs derived from bovine milk, mainly because they can be isolated with much higher titers than EVs derived from cell culture. In numbers, that means that according to the literature, about 300 milligram EVs per liter could be isolated. For those who are less familiar with extracellular vesicles, I'm just going to give you a brief overview on the main characteristics. Extracellular vesicles contain thousands of compounds, and they are composed of distinct classes of lipids, which are characteristic for EVs. Importantly, these lipids are arranged in an asymmetric manner in the double layer membrane of the EVs, which is shown on the bottom lower left graph here. Furthermore, many different proteins, small molecules, but also nucleic acids of different nature are present in the EVs. So they do not come empty, are hugely complex, and their composition varies a lot depending from which source they were isolated. Now, in our hands, we adopted a purification protocol for EVs based on fresh milk that was published in Cancer Letters in 2016 using consecutive ultracentrifugation methods, like shown here in the upper part of the slide. However, we quickly realized that this protocol did not yield sufficiently pure EVs. We did proteomics analysis, and we identified a large number of non-EV proteins, and that led us to improve on the published procedures, which is shown underneath, by adding another density gradient, and in that way we were able to isolate about 50 to 100 milligrams of purified EVs. Now, by using electron microscopy, we could demonstrate that there is less background debris present in these purified EVs. And further structural investigations using cryo-TEM also demonstrated the presence of these lipid double layer membranes as expected for such lipid-based vesicles. We then carried on and did the physicochemical characterization needed to understand and characterize the particles better using dynamic light scattering, like shown on the left, which gave sizes of about 190 nanometers, a polydispersity index just underneath 0.2, and the negative theta potential. And importantly, we could also demonstrate the high alpha helical content, like shown on the right-hand side of the graph, due to the nature of mainly tetraspanins, which are EV markers present in the extracellular vesicles. We then moved on and did used Western blot technology, and we were able to identify typical EV markers like TSG101 and also CD9. And preliminary immunochem confirmed the presence of these EV markers on the surface of the vesicles. We finally did proteomics analysis of these highly purified EVs, and we found at least 223 proteins, out of which 518, or about 63%, belong to the class of extracellular exosomes, according to the ExoCarta database. We were also able to demonstrate, like shown in this volcano plot in the middle, the removal of non-EV proteins by additional gradient ultracentrifugation, like, for instance, A2M, ApoA2, and HBD, which is hemoglobin, which are all protein of extracellular origin and must be considered as a contamination of EVs if not removed. Lipidomics analysis identified 8 major lipid classes, and within these, more than 200 individual lipids could be proved by mass spectrometry analysis. And once the physicochemical characterization of these purified EVs gave us the satisfactory information, we started to develop procedures for loading them with LNAs. And the best methodology that we could find was a so-called squeeze technology, where the EVs are pressed through a 100 nanometer membrane in presence of LNA solution, which then results in a quite low amount of loading into the EVs of about 2 to 5%. Now, in order to increase our confidence that the LNA was indeed in the interior cavity of these exosomes, we treated the loaded sample with a solution of detergent, which is shown on the right bottom part of the graph. And after the treatment, the LNA then became visible on the agarose gel, whereas in the absence of the detergent, no free LNA band was observed. So this gave us quite high confidence that indeed the LNA was not just attached to the outside protein corona, but rather accumulated inside the vesicular structure. Subsequent physicochemical analysis of the LNA-loaded EVs then revealed that the overall PhysChem parameters were largely unchanged, giving us the confidence that the mechanical loading procedure did not damage the overall properties of these vesicles. And now we needed to assess them for oral bioavailability, which we did in mice. We used the validated MALAD1 LNA and orally administered the extracellular vesicles at 0.21 and 2 milligram per kg to mice. We used the hybrid ELISA technology to measure the exposure of the LNA in a variety of organs, including the liver and the kidney. And we were able to find about 1% of the total administered dose in the liver and the kidney, but none of the other organs we could find any hint of LNA above the LOQ. It is also to be emphasized that the amount that we found in the liver and the kidney was below the IC50 of the MALAD LNA, as we determined by separately on cell culture assays. Now, like I mentioned at the beginning of my talk, we were mainly interested whether we could recover a fraction of the LNA in the brain parenchyma after oral dosing, and whether this fraction would be able to downregulate a target gene of interest, which in our case is the long non-coding MALAD RNA. And as you can see from the graph depicted on the right, we could not measure PD in any region of the brain isolated. And the same is true for the liver and the kidney, where most of the LNA was detected. So in our hands, unfortunately, we were unable to confirm oral bioavailability like reported in some of the literature, at least not to the extent that was reported on a number of occasions. If there is bioavailability, it is quite low, which renders milk extracellular vesicles rather unsuitable to repurpose for the delivery of oligonucleotides to the brain after oral administration. And in summary, we were able to increase the purity of the isolated bovine milk extracellular vesicles, which lies in the area of about 1%. Our study, but also other reports in the literature raise a number of questions within a pharmaceutical environment, which is the huge complexity of these vesicles, if they were to be exploited as drug delivery systems. What are the major critical quality attributes, batch-to-batch variations from the starting materials, and many more. So there is still a long way to go if such exosomes are to be successfully repurposed and applied as specific drug delivery systems. And with this, I would like to thank all who contributed to the work, and I would like to thank all of you for your virtual attention, and I'm happy to take questions.
Thanks a lot, Mike. I think excellent story, and I think very important to bring out there, because it's very important also to share the negative results where things don't turn out as you expected, perhaps at first. Next one up is Kunut Al-Jamal. She's chair of drug delivery and nanomedicine at King's College in London, and she will be our next speaker, talking about exosomes as pancreatic cancer therapy.
Okay, so thanks, Ray, for the introduction. So today I'm going to talk to you about exosomes and their applications for pancreatic cancer therapy. I'm afraid I won't talk about the detection, mostly because of the short time of the presentation. So I'll kick start. So we have targeted one important protein called PAC4. PAC4, or the PAC family, has been indicated in the progression of pancreatic cancer, and it has been shown through the use of chemical inhibitors that you are able to delay the growth of subcutaneous xenograft models in animals. So what we would like to do is move on to the use of biologics to treat pancreatic cancer through inhibition of this PAC4 target. We were interested in exosomes for the obvious reasons that my colleagues have talked about. So the work was mainly conducted by Lizu Tzu, a Marie Curie fellow, and he has generated very important data that I'll share with you today. So we hypothesized that if you're able to deliver successfully high concentrations of siRNA for PAC4 and successfully knock it down, you're able to delay the tumor growth of the animals and improve their survival. However, preparation of an effective model is not a trivial manner. So we have used our pharmaceutical science expertise in designing these systems in the best way possible. If you work with exosomes, you may know also that culturing them, isolating them when it comes to having different types at the same time is challenging. So we employed what we call design of experiment approach, which allows us to minimize the number of experimental runs, as well as look at the factor-factor interactions. So the way it works, you select your variables. So you have the cell lines, you look at different exosome types, and you look at the effect of time and dose as well. So this is what we call the level. So we have to set up a minimum and maximum. For example, in terms of dose, we go with what range we would like to do, the time points that we would like to study, and combine the different parameters, which if I tell you, for example, rather than having 160 conditions, you may end up having just 35 conditions. That then allows you to draw some conclusions. So we culture these exosomes in what we call bioreactor flasks, and these bioreactor flasks allow you to put more of these cultures in your tissue culture incubator. So rather than having 15 big ones, you can just have one, and you end up having a concentrated volume that then again reduce the processing time. So going through the stepwise centrifugation, and then we isolate the exosomes by ultracentrifugation over sucrose cushion. So you have the exosomes, we characterize them. So in terms of concentration, we're able to get this number, 7 times 10 to the 11 from one bioreactor flask, enough to do several studies, including in vivo studies, and the size was within what we expect. The exosome P particle to protein ratio was above 3 times 10 to the 10, which is considered as pure enough to proceed. This is just an example of the PANC1, but we've done the characterization for all these exosomes. And then we would like to actually be able to do dose response, know how much is taken up. And for that, we employed copper-free click reaction, where you utilize the amines on the exosomes with the DPCO conjugation, and then you're able to alter the probe you want. So you could have a a near-infrared probe, green, red, chelating agent, whatever you want. So we published this very recently in Journal of Extracellular Vesicles, and the information we have is just listed here in terms of labeling efficiency and how much we're able to recover, which is about 80%. So then what I show you here is actually the correlation between the observed uptake. So on the y-axis, you can see the number of particles taken up in cells, and on the x-axis is what you predict. So what you can tell is you're able to find a correlation between how much you take up versus the predicted, which means you can use this equation later on to actually input any numbers that allows you to know if you add X exosomes from this source into this cell line, this is how much you're going to get. And that shows you, for example, here we are drawing a comparison between three different types of exosomes isolated from PANC1 or melanoma or other types, really to see the relationship between the uptake of the exosomes and the cells. And what I would like you to look at here, the numbers in the circle, for example, if you incubate for 25 times to the 10 particles for 24 hours, you're going to get this number. And that's the prediction model we would like to see. What you can see is you have higher uptake of exosomes of PANC1 into their mother exosomes, into their mother cells. So that allows you to actually select the type of exosomes we would like to take to insert our siRNA in. So these are quantitative data to show the uptake with time. Interestingly, if you look at uptake of PANC1 exosomes into PANC1 cell lines, you can see there's no saturation and there's continued uptake, while if you mix other type of exosomes with the cells, you have some sort of leveling in the uptake. We still have to draw a conclusion why this is happening, but the prediction is it seems PANC1 exosomes utilize multiple uptake mechanisms into the cells, and that's why it doesn't get saturated as when you have other mix and match combinations. So we labeled them with a CY 7.5, and we looked at the uptake into the tumors. And what I would like you to see here is the picture in the bottom right, where you can see the uptake in the tumors. You have the free label CY 7.5, and you can see actually the dye exhibit a different profile from the labeled exosomes. So that the dye distributes everywhere in the body. You can see from the top image, while the exosomes have localized distribution clearly in liver, spleen, lung, as well as the tumor. So this is quantitative data. So if you look at the inset here, you can see the uptake of the different combinations. So you have untreated or the dye alone or the exosomes, and you can see the left and right tumors. You can have constantly higher uptake of the exosomes compared to the free dye. And these are PANC1 exosomes in PANC1 tumors. If you look at PANC1 exosomes in B16, F10, which is quite highly vascular compared to the PANC1 xenografts, you can see you don't have this pattern. So even in vivo, we have this affinity of PANC1 exosomes to the tumor, which is quite interesting. So it's beyond the passive targeting. There's something to do with internalization as well. So we electroporate these exosomes to allow encapsulation of siRNA, and we get approximately 14% encapsulation efficiency, sufficient enough to deliver a therapeutic dose of siRNA. You can calculate you have about 30 to 80 siRNA copies per one exosome. These are confocal images showing the uptake of both the labeled exosomes in green and the siRNA in white, and you can also observe siRNA in a non-encapsulated matter. So again, there's lots of work to do to understand the release kinetics. Looking at efficacy in terms of silencing, so our protein, which is PAC4 here, you can see silencing efficiency is comparable to the lipofectamine, both in Western blot and in using flow cytometry. And the comparison you can draw is lipofectamine is a cationic agent used commercially. However, these liposomes are non-cationic and are still able to protect and deliver functional siRNA. So looking at the migration, so we use the in vivo scratch assay to see if silencing of PAC4 is sufficient to actually delay tissue migration into the wound area. And the siRNA is a negative control here to eliminate toxicity or functionality of the cargo from the exosomes. And we can see, for example, with time, the wound start filling itself with the cells in the untreated, you see over time. However, when you have the siPAC4, you have reduced migration. That's what we really want. We want in vivo to reduce the migration of the cells and therefore metastasis.
Sorry, could you round off?
Yeah, okay, so we looked at the therapy and you can see we have improved delayed tumor growth in these mice as well as improved survival. You can see huge silencing in the PAC4 as you see with the light stained area compared to the negative control. And there is also no toxicity or changes in the body weight or alteration of liver enzymes. To the contrary, you have the PI, which is a positive control that, although it shows you the similar efficacy, we observe elevated liver enzymes showing liver toxicity. So in conclusion, we have developed a way to modify these exosomes where we are able to track them and establish a dosimetry profile that allow us to choose the best candidate and confirm that actually siPAC4 is a good target to treat pancreatic cancer. This was done with intratumoral administration as a proof of concept, and there's a lot more to be done here. So I would like to thank the team and the people who contributed directly to this work, mostly Lisa, who did most of the experiments, as well as our basic science and clinical collaborators, and thank you for listening.
Thank you very much. Excellent presentation and powerful therapeutic effects. Really interesting to see. We go straight to the next lecture by Dmitry Simberg. He's working at the University of Colorado, and he'll talk about the synthetic counterparts, the liposomes.
Okay, hi everyone. Thanks for Beato for organizing the meeting. Palmar-plantar erythema or hand-foot syndrome in patients, which is causes quite a bit of suffering to the patients. So let's see how I switch the slides. Okay, so it's caused by extravasation of the drug. Definitely, doxorubicin comes into skin tissue and severe inflammatory reaction and severe pain and all the symptoms in the palms, feet, and the areas of pressure, hence term hand-foot syndrome. It's up to 50% of patients and major side effects leading to dose reduction. So what's important is the mechanism of this accumulation is not well understood, and there we kind of get interested in this through in a totally different project, but it's kind of like now one of our interesting research topics. So if we inject doxil, or basically there is now a generic drug, lipodox, you can get from a hospital pharmacy. If you inject into mice, you see that there is pretty long half-life, about 30 hours half-life. And you can clearly see with the low magnification microscopy, there's excessive accumulation of doxorubicin in the skin tissue. And if you wait long enough, mice start developing necrosis, and there's like this necrotic nails and erythema. So there's quite a bit of this toxicity observed in mice, which is kind of similar to humans. So the PK of doxorubicin was studied before. Terry Allen published a paper 2003 showing that there is a buildup of doxorubicin after injection of pegylated liposomal dox in the skin, and foot pads kind of have a very fast kinetics as opposed to the rest of the skin, similar to basically feet and palms in humans, like basically in the areas of pressure, I guess. So if we inject doxorubicin and stain it with different markers, we see that doxorubicin accumulates in vimentin positive cells, which are mostly fibroblast and dendritic cells. And there is some accumulation in the immune cells, CD45 positive cells, and there is some colocalization with CD31, which is a vascular marker. So if you look at the CD31, you can clearly see that some of the doxorubicin is outside of the blood vessels. So there's clearly an extravasation of the liposomes going on, and doxorubicin accumulates in the nuclei, making them like a purple-like color. So we decided to study kind of skin accumulation, more dynamic process in the living animals. So we designed this instrument based on fiber optic, which is provides infrared light, infrared source, and you can collect the light with this fiber, and it reads the spectrometry, and you get pretty much integrated in emission over time. So so you just basically put a mouse, you inject with the liposomes or whatever nano-drug delivery system, label it with infrared dye, and you see that you can shine light on the skin and collect the signal over time. So we used pegylated and non-pegylated liposome labeled DIR. It's like a lipophilic, well-known indocyanine dye that's in the near-infrared spectrum, and you can see that pegylated liposomes show higher accumulation over time when liposomes are already gone from the circulation, but pegylated liposome reside in the skin, and there's probably around 5 to 7% of the injected dose per gram skin accumulates over time. If we look at the biodistribution of the liposomes, so DIR labeled liposome, after several days, you see that intestine, spleen, kidney, liver have something, but lung, heart, and brain don't have much. And the skin shows dramatic accumulation, especially in the paws and the pads, walking pads in the mice dissected, but also in the back and the abdomen. There's like very interesting and strange structures, periodical structures in the back of the mice, as well as the diffused accumulation of the skin. So definitely skin accumulation is one of the predominant for liposomal accumulation in the body. So we decided to go further and try to look at this imaging, doing intravital imaging of accumulation of the liposomes. And for this, we would need we would inject mice with liposomes either with doxorubicin inside, which is itself is fluorescent, or dye. It's a visible red dye that's also lipophilic and stains. Incorporates in the membranes. So we prepare the skin flap, and basically mice can be mobilized and imaged. So we inject with liposomes, and then to highlight the blood vessels, the nuclei, we injected fitsidextran, and we imaged the mice for some time period. And these are dye-labeled liposomes. You see at 30 minutes, they are kind of accumulating mostly in the blood vessels. But about 2 hours later, up to 7 days post-injection, we start observing numerous cells that accumulate liposomes outside of the blood vessels. So it really kind of jumped the endothelial barrier and kind of crossed them and extravasate. And doxil looks like it also appears like it's dispersed staining outside of the blood vessels in the skin. So definitely there is an extravasation process of liposomes going on. So we thought, what if we injected doxil together with another liposome that's labeled on membrane with a different dye? So we prepared, so we used doxil, which is 88 nanometer liposomes, zeta potential minus 280 millivolts, and we prepared a similar composition and size liposome labeled with the dye, far red dye D. 102 nanometer, a little bit less negative. After the injection of both liposomes, we mix them the equal amount of liposome, inject them in the mice after 1 hour with high magnification microscope, and this is 200 nanometer bead. You can see these liposomes kind of staying within the size range. They're not growing, and they're still in the circulation. The circulation time of both liposomes appear pretty similar, although the ideal liposomes seem to clear a little bit faster. So we looked at 1 hour accumulation of skin, and there we find an interesting result that looks like the ideal liposomes spreading and accumulating and extravasating in a tumor are much more efficient than doxil. If you just look at the both types of fluorescence, this is as soon as 1 hour post-injection. Here is the magnified image showing the blood vessel. There is tons of these vesicles, kind of a dotted pattern that's extravasating the blood vessels. And some of them colocalizing, but some of them not colocalizing. So it looks like DID and DOX going through some sort of separate pathway in extravasation. If we look at many microscopic images, we can see that indeed the DID covers significantly larger area than doxorubicin. So it looks like these two liposomes going in different pathways. So it made us wonder, what's the mechanism of this accumulation, this difference? If we had a pore gaps in the endothelium in the skin, we would expect both liposomes to leak out approximately to the same extent. But we thought, okay, if there is some sort of transport pathway, this doxorubicin and DID sorted in different pathways, and that's why we observed these differences. So just to kind of have a high magnification resolution images of what's going on, we prepared liposomes with a different lipophilic dye that has an amino group. And why? Because these dyes are not fixable usually. So presence of the amino groups allows fixation of the tissue. And then we can prepare like cryosection and do very high magnification images. And what we see with the tumor, with the skin endothelia is that these are red blood cells inside. Like they are a little autofluorescent, but you see endothelial cells are filled with the dye, with the samine dye, and you see those kind of vesicles budding from the endothelial cells, which strongly reminds us of this sort of EV sort of formation process that happens sort of in vivo. We went back to the literature and tried to understand really what's the process behind this. And there is a pioneering studies done by Harold Dvorak, who discovered VEGF and Dvorak from Harvard. They basically describe those structures called VVO, vesicle vacuole organelles, that's operate in the endothelial, in tumors as well in the skin. This kind of like this vesicular structure that span the entire endothelial cells. And they look like they're larger than caveola. Caveola has really a limit of size, about 50 nanometer for nanoparticles to move across cells. But the VVOs seems like they're larger and could really accommodate our liposomes. So we did a TM analysis of the skin.
We need to round off, please. Yeah.
Yeah, so the TM analysis doesn't show much accumulation of the liposome, but it really showed the extensive vesicular trafficking. And just to distinguish between caveolae process and a different type of process, we injected Cy5 labeled albumin, and we see that they don't colocalize. So dialiposomes just kind of going through different pathways. So in conclusion, drug carriers accumulate, causing toxicity, and we can monitor that. And the extravasation for blood vessels strongly resembles extracellular vesicle mediated transcytosis, but we're still working and trying to prove that. And we really think that better understanding can improve safety drug delivery system. So I want to acknowledge the funding from the NIH and our school, as well as my students, Lara Lottie and James Griffin, who did most of the work for this study. Thank you very much.
Thanks a lot. Interesting observations on the fate of liposomes in the skin. Thanks. We continue with Dr. Wafa Al-Jamal, and she's going back to a sort of mixture of the two exosome mimetics, and she's reader in nanomedicine and drug delivery in Queen's University in Belfast.
Many thanks for the introduction. It's a great pleasure to be here today to talk about recent work targeting metastatic prostate cancer using exosome mimetics. Prostate cancer is considered the most common type of cancer in men and worldwide, where expected 1 in 8 men will have prostate cancer at some point in their life. It has also the highest mortality rate after lung cancer in men. Normally, patients will be treated based on the stage of the disease, as we see here, stage 1 and 2, where normally the prostate cancer remain localized within the prostate tissues, and patients respond well for radiation, surgery, and hormonal therapy. However, over time, stage 3 and 4, we start to have metastasis, where the cancer starts spreading outside the prostate, mainly to the bone, and the only way to treat patients mainly currently with the chemotherapeutics, where we have successfully increased patient survival by a few months, but patients still suffer from basically the side effects. Therefore, there is unmet need to deliver new therapeutic approach or strategies to deliver chemotherapeutic to metastatic lesions with high efficacy and reduced side effects. Prostate-specific membrane antigen, the PSMA, is a transmembrane glycoprotein. We have heard a lot about PSMA in the last few days. It has an extracellular domain which recognizes targeting ligands. It's overexpressed on prostate cancer cells and the new vasculature of solid tumors, but not on healthy vasculature. It's also upregulated upon androgen deprivation. Several targeting ligands have been engineered, like peptide, aptamers, and the most clinically relevant monoclonal antibodies, where they have been already administered in patients to deliver cytotoxic drugs or radioactive isotopes for imaging or therapy. These targeting ligands also have been incorporated in different nanotherapeutic platforms for imaging and therapy. We heard a lot about exosomes and the benefit of using exosomes due to their biocompatibility and intrinsic affinity, but also we heard about the exosome biomimetics, where they are slightly similar to the exosome, but we develop them in a way to overcome some of the issue like poor yield and poor drug loading. In the current work, we aim to bioengineer PSMA-targeted exosome mimetics to target metastatic prostate cancer, which hopefully could help us to deliver more effectively and safely therapeutics to metastatic lesion in patients. What we did in this project, first we had to bioengineer PSMA-targeted cells, then use them to prepare our targeted mimetics, followed by in vitro and in vivo characterization using different prostate cancer models. To start with, we selected U937 cells, and we wanted to express a short anti-PSMA peptide on the surface of these cells. So we constructed a plasmid to guarantee the expression of the peptide on the surface of the cell membrane, and we also incorporated H-tag protein to enable us to follow up on the expression of the peptide. Sorry. We use two different ways to deliver the plasmid, either lentiviral or nucleofection, and then the transfected cells or genetically modified cells, we refer them as U937L for lentiviral transduced cells, or N for nucleofected cells. Following that, basically the characterization using Western blot, we detected the H-tag expressed in the transfected cells. We have high level in both cells compared to the wild type, which indicates successful expression of our peptide. And this was also confirmed using PCR with high level of the PSMA peptide mRNA. Then we wanted to start fabricating our exosome mimetics. So we extruded the genetically modified cells through the different size pores membranes, and we used the size exclusion chromatography to purify our vesicles. We looked for different markers. The FLA1 is expressed on the monocytes as well as the exosome markers to make sure during the extrusion we still have good level of protein incorporated in the cell membrane. And we compared the exosome mimetics to the original cells, and we could see all the markers are still present. We also looked for the H tag, which indicates the presence of our peptide, and we can see high level as well compared to non-transfected cells. This uptake study using C4-B tumor model, which overexpressed the PSMA. We fluorescently labeled our vesicles, and we could see following 3 hours of incubation, we have significantly higher uptake in the targeted system compared to the non-targeted. So the results were promising in terms of the peptide. It seems it's working. It increases the uptake in the prostate cancer cells. However, due to the limitation of poor drug loading with this system, as our results showed, as well as others, we tried to modify it a little bit to make sure that we are designing a system with high good drug loading that will enable us to using it in the future for therapy. So we fused the hybrids with liposomes, and in this case, we had a lipid film. We hydrated the lipid film that we use for liposome production with the cell-derived membranes, and then we followed up with our production exactly as with the exosome mimetics. And our hope is really to increase the drug loading inside while maintaining all the targeting ligands on the surface. So we characterize these hybrids basically whether they have a drug or not. And using NTA, we could see the size was still below 150 nanometer. They have really comparable size, which indicates the presence of the peptide doesn't really affect the property of the hybrids, as well as we could achieve high drug loading. We use here a DOX-PSA prodrug, and we maintain around 60% loading, which looks promising for further studies. We repeated also the characterization for the exosome and anti-PSA peptide expression, and again, we can see all markers were present in the hybrids. And under transmission electron microscopy, we could see basically all the hybrids, whether targeted or non-targeted, they had the same shape and morphology and size. Then we move towards our in vitro characterization. So we start using different prostate cancer cell lines. And as we see here, we use the PC3, which is PSMA negative cell line, and the C4-B and the LNCAP, which both express the PSMA. The C4-B represent the late stages of the disease, the hormone-independent cell line, while the LNCAP represent the early stages of the disease. We labeled our vesicles using dye, and then we looked for the uptake using flow cytometry. In PC3 cells, we could see nice uptake, which is time-dependent with the non-targeted vesicles. However, surprisingly, we noticed with both targeted systems, significantly lower uptake was observed, which we need to investigate this further. We don't know the reason behind it, but it was very obvious in the PC3 cells. With the LNCAP and C4-B, we could see significantly higher uptake with the targeted system compared to the non-targeted, which again confirms our hypothesis by increasing the selectivity to prostate cancer cells by incorporating the PSMA peptide or anti-PSMA peptide. These studies, they were carried out in serum-free media. So to make it more physiologically relevant, we repeated the same experiment with the C4-B using 10 and 50% serum. And we could still see higher and significant uptake with the targeted system compared to the non-targeted, which looks very promising for both in vitro and in vivo studies. Since we could see the uptake was slightly higher and maybe faster with the hybrid system prepared using the transduced cells with lentiviral, we decided to select this system for further in vivo studies. So what we did next, we labeled our hybrids with a near-infrared dye, and then we intravenously injected the hybrids in C4-B tumor-bearing model, which is PSMA positive. And we could see significantly higher uptake in the tumor with the targeted system compared to the non-targeted. That was not the case once we repeated the experiment in the PSMA negative model, which indicates the presence of the receptor. The PSMA receptor is really important to target for the uptake of these exosomes in vivo. Then we tried to see if we can achieve also targeting in metastatic prostate cancer models. So what we did, we established a luciferase expressing C4-V model. And this metastatic model, we could see it localized mainly to the bone and also to the abdominal area of the mice. Following 24 hours of administration, we could see nice co-localization between the fluorescence signal on the right with the bioluminescence signals in the knee area where we see the metastasis, as well as we could see some soft metastatic lesion in the abdomen, the bottom image here. We could see the nice co-localization between the two, as well as in the lung in one of the mice. We have the metastasis to the lung, which co-localized with the fluorescence signals, which indicates basically that the targeted system is also targeting metastasis in vivo. So our conclusion that we managed to prepare a bioengineered stably expressing anti-PSMA U937 cells via nucleofection and lentiviral transduction. We also showed that the targeted exosome mimetics, they have high affinity to PSMA expressing cells in vitro, as well as increased tumor accumulation following systemic administration in both solid and metastatic C4-V cells. And in this case, what we would like to do next is to evaluate the therapeutic efficacy of our drug-loaded exosome mimetics in metastatic prostate cancer models. So in conclusion, I would like also to thank the team who did the work, mainly Maya for carrying out all the work and people helped with the in vivo studies and the funders, of course, and thank you for your attention.
Thank you very much. Great presentation. Thanks.
Yeah.
And the final presentation for this session is by Jose Caballero and Gerald Ria. And they will talk about polymeric bile acid nanoparticles.
I see that I was on mute, sorry. I wanted to say thank you to everybody for Beat, Patrick, the organizers of the cleanup, from the perseverance and resilience to maintain this excellent meeting, and for giving us the opportunity to communicate our work. So this was a double presentation, but we decided to make it short, and I will give a brief introduction of type 1 diabetes, and then I will leave most of the time to my colleague that will present this new oral nanoparticle platform. So type 1 diabetes is an autoimmune disease that is characterized by the destruction of the insulin-producing beta cells. The disease is described as a continuum progression from an asymptomatic genetic predisposition to a long-standing disease that requires exogenous lifelong insulin for survival. And there is not consensus on what is the initial event that will trigger the disease, and the current hypothesis suggests that there is a cross-reactive event to an infectious agent that will awake autoreactive T cells. The disease can be unadverse for many years. However, what is clear is that it can be predicted once is detected a series of autoantibodies to the pancreatic antigens. And the current practice is to declare the disease when the patients display an abnormal glucose level and all the classical symptoms that are associated to type 1 diabetes, and that they are associated with a significant beta cell loss of beta cell mass. From then on, there is not much hope, and basically what everybody needs is to have lifelong insulin with all the problems of compliance and with all the side effects that are associated to the treatment. However, the disease, as I mentioned before, starts much earlier, and there is a possibility to treat these early stages even in a prophylactic manner in individuals that they are at risk. I'm having some technical problems in my screen that is showing something, but I continue. And there is no cure for type 1 diabetes, and most of the therapeutic perspective relied in broad immune suppression, as is happening also for many other autoimmune diseases. But very recently, there was a clinical trial using an anti-CD3 monoclonal antibody, the Liposomes, which was administered on 14 consecutive days through an IV infusion, and it generate a significant delay in the time that the people transition to the overt disease. This therapy is further explored, and the idea is to have a subsequent repeated course, 6 months maybe after the first one, but we still need to see if this will maintain the disease for a lot longer. Immune tolerance approaches, on the other hand, they have more hope in terms of having a greater specificity and associated safety profile. However, all of these therapies that they also generally involve injected products, they can be IV or they can be even intra-lymph nodes, they have not succeeded so far to achieve the expected endpoints. And the other thing is that I believe that now many people start to consider that there is a possibility to induce immune tolerance, but the maintenance of immune tolerance will require to have a kind of repeat boost of the tolerogenic antigen. So in summary, there is an urgent medical need for therapies that they will target the root cause of the disease and that they will target all the patients at all the different stages, and particularly they should be using patient-friendly treatments. And on this point, I would like to pass the virtual floor to my colleague Gerald, that will tell you how can this be achieved.
Hopefully, we have high aspirations. Today, I'm sharing a new nanoparticle platform with you that may solve many of the issues that are impeding better treatments for diabetes and also autoimmune diseases in general. This nanoparticle's intrinsic properties overcome the obstacles of delivering biologics through the oral route, through the GI, and it offers its own therapeutic effects, which synergize with the cargo. Our flagship product is a bile acid that has been polymerized. We're using ursodeoxycholic acid, and we make a polymer of approximately 7 to 10 monomers out of it. And then we go through a simple emulsification in the presence of drugs or molecules that will act as the cargo and are entrapped in the solid nanoparticle. The work that I'll share with you is with a nanoparticle that is a nominal diameter of 350 nanometers with a negative zeta charge. And the base monomer, ursodeoxycholic acid, was chosen for the synergies with metabolic diseases, inflammatory diseases, and because it's already an FDA-approved drug. The manufacturing process is pretty simple. It's a single or double emulsion process. We can put in multiple cargoes at the same time or single cargoes. We have looked at dyes, small peptides, proteins, immunomodulators, and are currently working on monoclonal antibodies. We have achieved loadings of 10% and loading efficiencies above 50%, and are currently scaling our production from 100 grams to 1 kilogram GMP. The work that you'll see has been administered to rodents as a solution by oral gavage, and in the large animals in a capsule. And we have not done any stability testing to date. One of the interesting aspects of this nanoparticle is that at the 350 nanometer diameter, the biodistribution is tilted toward pancreatic localization. And so on the left side of the screen there, you can see PLGA and then different polymerized bile acids. The one on the bottom left-hand corner is polymerized ursodeoxycholic acid. You can also see that we have achieved roughly 25 to 40% bioavailability in rodents. The lower bioavailability is the 350 nanometer product. The higher bioavailability is with smaller nanoparticles. We can also change our biodistribution and localization based on nanoparticle size. As you can see, as the nanoparticle goes down in size, we get a much higher accumulation relatively into the spleen. The characteristics of our nanoparticles, they protect the cargo in the GI with very little leakage. This is similar to a enterohepatic or enteric coating on drugs that just naturally happens with our nanoparticle. And then the PUDCA enhances both passive and uptake, active uptake through the intestine. So we are piggybacking on the enterohepatic recycling system in animals. And then we also are a weak agonist, TGR5 agonist, which triggers anti-inflammatory and metabolic properties as well. So the active part of our transport is through macrophages. They endocytose the PUDCA nanoparticles preferentially, and then are skewed away from activated pro-inflammatory M1 types toward M2s. We promote anti-inflammatory cytokines and regulatory T cells, attracting chemokines by antigen-presenting cells. We induce secretion of GLP-1 from the intestinal L cells, and then in turn stimulate insulin release from the pancreas. We have looked at diabetes treatments in a number of rodent and swine models. In the spontaneous model, you'll see on the left the effect of a rather large amount of the empty particles, as well as insulin loaded in the particles. And you can kind of think of that graph on the left as the short-term effects. And you can see that we obtained normal glycemia with the insulin-loaded particles over the course of the dosing. On the right side of the screen, you will see the chemically induced cyclophosphide model, and using rapamycin to prevent the destruction of the beta cells with the Tregs being increased by a significant amount. And then we took that learning forward and looked at longer term antigen-specific tolerance and how to restore the diabetic animals in a much safer manner. And so this is using an immunomodulator co-encapsulated with a pancreatic autoantigen. That's what you see as the red line on the graph. The green line is with the nanoparticles with only the pancreatic peptide in there. And then the blue line is our empty nanoparticles. And what you can see is we produce, we achieve normal glycemia for about 60 days with both formulations with the autoantigen, and that we achieved almost 90 days when we added the immunomodulator. And our next steps are to look at repeated dosing and to keep the animals in a continuous remission. We have also shown antigen-specific tolerance in other autoimmune diseases, including multiple sclerosis and rheumatoid arthritis. For late-stage diabetics, after the beta cells are already destroyed, We have looked at delivery of exogenous insulin. And on the top of the screen, you will see the release rate of insulin and neutral pH on the top left. And then you will see a stepwise graph with the first 2 hours and a pH 2, and then a neutral pH. And as you can see, we get very little release of insulin into the low pH environment, and almost all of it is released into the neutral pH. And we have achieved 85% plus bioactivity of the released insulin. The bottom graph, you can see these are moderately diabetic Ossabaw swine that have been treated with aloxan to chemically induce the diabetes and then stabilize for 2 weeks. We treated the animals over the course of 7 days and got very close to achieving normal glycemia within 7 days. The insulin that we delivered in this experiment was roughly 1 unit and roughly 300 milligrams of the PUDCA nanoparticles. So we are looking at diabetes holistically, and would like to have treatments that not only act as a prophylactic, but also treat the disease during its progression, and then at the end of the disease in late stage diabetes, deliver exogenous insulin. And so what we have is a nanoparticle that protects the cargo from denaturing cleaving in the GI, enhances uptake due to enterohepatic recycling, basically biomimicry. It delivers the cargo, and as I said, we've done small, medium peptides and proteins, and now we're working on large monoclonal antibodies. We can vary the PK profile, release profile, by changing the density or the fabrication method. And then in diabetes and autoimmune diseases, the intrinsic bioactivity actually acts in a synergistic manner with the cargo as a weak multivalent agonist to TGR5, giving us anti-inflammatory cytoprotective effects. And then we're also exploring the NF-kappa B synergies with immune modulators such as rapamycin. Thank you guys very much, and I look forward to any questions or discussion.
Okay, great. Thanks a lot. I see that some questions have been coming in, and also in the panel chat there have been some questions that we can also address plenary. Okay. Oh, sorry. Let's first go to the question about the last speaker. So Joy Wolfram, who we'll I think know, asked a question about how came this idea about? So how did it all start?
The PUDCA nanoparticles?
Yeah.
All right. So that all started from looking at honestly the findings of in a lot of Asian-based medicine from bare bile acid. That was kind of the initial underpinnings. Then a lot of studies started to come out with bile acids used as targeting moieties for nanoparticles, and what we found, and Dr. Tariq Fahmy was really the inventor of this technology over at Yale, is that you could actually increase the not only the therapeutic effects, but also the bioavailability and other things if you made the entire nanoparticle out of the polymerized bile acid. And so it kind of started out looking at a metacarrier and trying to marry the effects of ursodeoxycholic acid with a nanoparticle. And then a lot of happy happenstance occurred along the way. And, it ended up that we were able to protect cargo, deliver it, and that it actually synergized even better than the base monomer. So really it started out from the idea of that bare bile acid, which is kind of a horrible thing, but it's it's a it's a long used medicine and folk medicine basically.
Interesting, thanks. Going to one of the first lectures given by Ivane. So one of the questions in the chat was, how do you dose, and also how often do you think you need to administer to maintain efficacy? Was one of the last questions.
Yeah, thank you very much, Ralph. This is actually a good question. So in our case, in order to be able to compare the efficacy of the extracellular vesicles that we were using to the free protein that is used in the clinics, we were dosing regarding the amount of the mass of the GLA. But in our extracellular vesicles, the good thing is that we have the loading is around 5%. So we can make a quite quick calculation and see that we need to administer like 700 micrograms of the protein loading of the extracellular vesicles to be able to see the efficacy. We haven't done so far any dose ranging studies. So we were dosing with the same doses that were used in previous preclinical studies with the free protein. So we don't know if we are able to reduce that dose, but we are pretty sure that we could go for that. So actually the next experiment that we are planning is that one, to reduce the dose and see if we can have the same effect.
Okay, thanks. Mike, a couple of questions for you. One of them is, how do you explain the discrepancy between what has been reported in literature and your own results?
Yeah, I think there's a number of factors there that can play, and of course it's different people doing the work as well. So it's very difficult to really say. And I think what is one of the fundamental problems with these type of studies is that often people use fluorescently labeled EVs or cargoes and then determine their fates and even quantify the payload or what they think they are the payloads using a fluorescent microscopy. And I think we know now that this can be dangerous, can be misleading as well. This is one of the factors, I believe. And purity, I mentioned that. Whether this interferes or not is very difficult to say. And also the loading type of technology, as you know, plays a role that some of these technologies are more suitable to load the nucleic acid into the interior side of the exosomes. Others are less suitable. So it's multiple factors interfering. Maybe it's just all together that makes the difference between what we have seen and what other people have seen.
Yeah. One additional question. Again, Joy Wolfram posted a question. What type of milk did you use? And well, the other question, of course, relying on that is would it matter?
Yeah, it's a good question. Actually, we thought about that as well, and we tried various sources of milk, but it was always Swiss milk. So, but then we took milk that was directly from a producer, from a cow, basically. But that was a bad idea because it was came along with a lot of milk bacteria, which we kind of contaminated throughout the whole process. It was very difficult to get rid of. So finally, we took pasteurized milk from a bio store somewhere in town and just stuck to the same origin of milk all the time, and that made the results much more reproducible. Mm-hmm. Whether it matters or not, we haven't really looked into many different sources, but what we have looked into is milk from different seasons, so from spring, summer, and fall. And we have also investigated whether the composition of the milk exosomes differs over the course of the year. And we were really kind of surprised to see that actually it was literally identical composition. So I think from that point of view, that was good. And other factors, we didn't really have the time to look into more in detail.
That's really interesting, because I remember that in the when we were using natural phospholipids for quite a while in the liposome field, and then the opportunity emerged to do mass spectrometry on the phospholipids from these natural sources like egg yolk and soy. And then we saw a clear difference on a composition between different batches from the same vendor, etc. So you open the whole Pandora's box by wanting to investigate that, because now all of a sudden this became an issue. But it's interesting to see that indeed the composition is largely the same. One question.
Yeah. I must say we were surprised by that very much. We outsourced the lipidomics analysis, so it's a completely unbiased result. So, but it was literally identical.
Oh, interesting. One additional comment also coming back to the composition. Did you compare the proteomics also before and after loading, for example?
No, we didn't. No, we didn't. We had limited access to the proteomics facility within Roche, and we could unfortunately not do that comparison. And I know this is a drawback, and I would have loved to do it, but you know, there's limits with every study you can do, and this was one of the limits that we encountered.
Okay. Thanks. One question for Dr. Simberg. The low signal of Doc's liposome, might that be due to quenching of the fluorescent signal? And more in general, I guess, how do you address these differences in labeling and in the fluorescent intensity, for example?
It's an excellent question. So we could be a possibility of quenching. So we did quite a bit of work to normalize the fluorescence of the indocyanine dye and doxorubicin, both in the mixture that we injected, also the calibrating the microscope channel intensity, as well as the imaging equipment, just to sort of to match the fluorescent mismatch. Doxorubicin was injected with about 100-fold excess over the dye-D fluorescence. So there is a possibility of quenching, but we did our best to match this kind of fluorescent intensities.
Okay, thank you very much. Question regarding the PEPCA nanoparticles. What are the key elements that contribute to the high accumulation in the pancreas? Question by Shanshan.
If you're talking about the key, mechanical properties of the particles themselves, you're really looking at the diameter and the density. So basically how you interact with the receptors in the acinar cells and the beta cells. But so you're looking at changing the diameter and the density and how you interact with those the exterior of those cells in the pancreas.
Okay, thanks. One question, and from the time, I'm not sure exactly which presentation, but the question is, how do you conjugate the peptide to the exosomes? And I think I've seen that in a number of different presentations. I think, for example, in Kulut's presentation, was it you did it by click chemistry, I think?
Yeah, I think we've seen it in some examples. Wafa showed something about the extrusion with other peptide expressing
let's it all.
So in our case, the chemical conjugation was through copper-free click reactions, which minimizes the step of purification. So you need to functionalize the amines on the exosomes, and then you have azide or functionalized moieties that you can also generate. But you can also use other amide coupling, etc. Mm-hmm.
Wafa, do you want to add something to that?
Yeah, okay, I'm just unmuting. Yeah, so basically what we didn't do chemical conjugation. That's why I said bioengineering. So what we did, we transfected the cell with the plasmid that would express the anti-PSMA peptide. So that will be expressed on the surface. And once we extrude the cells, the peptide will be already present. So we didn't do chemical conjugation. It was basically gene expression. Thank you.
Okay, thanks. Joaquin has a question for Kulut. Have you checked stromal cell activation by your PANC1 derived exosomes? Could they present deleterious effects in this regard?
So stromal cell activation.
Yes.
We haven't done that. No. It's a good idea to see if there is effect on even the extracellular matrix and how the rigidity of the matrix change. Something to consider for the future. That's a very good comment. Because the permeability was actually unexpected, considering how fibrous the tumors were, and we still saw high permeability. So that's a good experiment to do.
Indeed, an interesting observation, given the fact that indeed you expected almost to be impermeable to all kinds of nanoparticles, but you see this generalized tissue distribution. So interesting. Also a question addressed at me, actually, from Shiki Wang. Why are the extracellular vesicles much more potent than synthetic liposomes? Is it because of uptake? Is the donor and receptor the same cell line? We've seen it for a number of different combinations of EVs and acceptor cells. Although the efficiency of each combination differs, but we don't know exactly why that difference is there. So we do see that the same inhibitors work in the different cell lines. So apparently there's a sort of general uptake mechanism, but whether it's the increased uptake, whether it's reduced recycling that has been reported for the synthetic lipid nanoparticles, or whether it's improved endosomal escape, for example, by extracellular vesicles. These are things that we still don't know. We think we have the assay to examine it, so that will be indeed work for the next coming years, I guess. So yeah. That's it's an important area to try and understand where they separate or where the efficiency separates and how that comes about. But no clues yet. We only see the differences and no reason yet for that. Let's check if there are questions still. No, not online at least. Anyone from the panel members want to add any questions, ask any questions to each other? If not, then thanks a lot. I really enjoyed it. Nice presentations, and I want to close this session. Thanks all for joining, and I hope you liked it. See you hopefully next year in Basel.